Supercoiling in DNA is common and causes local changes in DNA topology, including changes in linking number. As we talked about in the last video, negatively supercoiled DNA causes a decrease in linking number. Most eukaryotic DNA is slightly negatively supercoiled. Positively supercoiled DNA has a higher linking number than relaxed DNA. Only really organisms which live at high temperatures, called thermophiles, have positively supercoiled DNA. This is because overwound DNA is less likely to denature due to the tight structure. Spirals can increase or decrease linking number depending on whether they are right or left-handed. Almost all DNA is packaged as spirals around histones. Normally, DNA is right-handed and forms left-handed spirals around histones, which contributes to the negative supercoiling of DNA. For DNA replication to occur, DNA must dissociate from histones. When this happens, there are more twists and less rides, which triggers DNA further downstream to also dissociate from histones. This diagram shows a step in the replication process where an enzyme called helicase splits the double helix in half, creating positive and negative supercoils. Topoisomerases are a class of enzymes which prevent too much positive or negative supercoiling by changing the linking number of DNA. Type 1 topoisomerases create temporary single-stranded breaks in the DNA which allow the uncut strand to pass through the gap, changing the linking number by 1. This is a passive process which doesn't require ATP. Sometimes, instead of the single strand of DNA passing through the gap, the DNA helix rotates around the unbroken strand, which also decreases the linking number by 1. Type 2 topoisomerases change the linking number by making double-stranded breaks in the DNA. The whole DNA coil can then pass through the gap before the break is resealed. This decreases the linking number by 2. In this example, the circular DNA loses a twist and a writhe when the helix passes through itself. All type 2 topoisomerases require energy from ATP hydrolysis. Type 2 topoisomerases are also responsible for catenating and decatenating DNA. Before cells can divide, DNA must be replicated by DNA polymerase, which is shown by the orange shape here. In prokaryotes, DNA is circular, so the two double helices produced will be linked together. In order for the cell to divide, they must be separated by topoisomerase, which is represented here by the green circle.